This is the component 2 taskbook, and we're on to the applications section, question 10. A lot of blurb to read. Question A1, the name. So the name of the work. The name of it is a Human Genome Project. The 100K project wasn't even thought of then, so this is early on. Possible abuse of this work is eugenics, so you should know what eugenics is, which is basically genetic engineering to improve the humans. The typical thing to say here is designer babies, but you need to sort of need to do a little bit better than that. So, encouraging desirable traits, discouraging the transmission, passing on to the next generation of undesirable ones. So if we say something like selective breeding of humans, an example of that might be, forced sterilization of, people considered to be feeble-minded or of an undesirable character, that sort of thing, or not the racial type that you prefer. So, e.g., sterilization of undesirable people with undesirable traits. Or you could just have, genetic engineering for the purposes of improving, babies, that sort of thing. So lots of possible answers there. More straightforward is, what is gene therapy? So just inserting a working copy of a gene into DNA, so that this working copy will be expressed rather than the faulty copy that was originally there. So inserting a working or new or correct or something like that, working copy of a gene or working copy of DNA to replace a faulty copy. Explain the function of liposomes. This is just telling a story nicely, this question. So, if gene therapy is inserting a copy, well, how do you do it? Well, quite often it's with these liposomes. You know about transport in and out of cells, and you know about plasma membrane, and you know about transport of substances through that membrane. Well, if it's lipid soluble, it'll get through that membrane easier than if it was polar. And so what these liposomes are kind of balls of lipids with our DNA in the middle. So those can fuse with membranes and use a kind of plasma membrane fluid mosaic model, and you can fuse and blend and that sort of thing. So much easier to get into the cell. So what are they? They introduce the new DNA into body cells. And it's just 1 mark, so we don't really need to give too much more detail there. Advantage of, and also disadvantage of using gene therapy. Quite often, if we're using these liposomes, they might correct cells that we have there, but cells replace themselves, and when they replace, they might replace from the original faulty cells, And so this therapy might need to be continuing for quite a long time. So possible reason it's not permanent. So we'll need retreatment as cells replaced by faulty cells. And whenever you're mucking about with DNA, there's always a risk that you might introduce something, a gene that you don't want. So introduction of other faulty genes, or DNA might escape. There's always the slight, you've got to bear in mind your DNA when you liposome, it might, get out, not into where you want, it might go somewhere else. You might, when you're mucking about with DNA, there's a risk of introducing, higher risk of cancers and things like that. But I've gone for a nice, kind of simple math there. 6 marks.